Hello, everyone. Welcome to today's live broadcast, Optimizing Reproducibility in Animal Studies Through Assurance of Animal Welfare. I am Brenda Kelly-Kim, and I'll be your moderator for today's events. LabRoots.com is the leading scientific social networking site, and we are proud to bring you this interactive web seminar. For more information, please visit us at www.labroots.com. Here's how this presentation works. We want to hear from you. Questions, comments, and even answers can be submitted via the green Q&A button at the lower left of your screen. We'll try to get to everyone, but if not, we'll make sure to follow up with you via email. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you can't hear or see this presentation properly, please let us know by clicking on the support button at the top right or the Q&A button in the lower left. Also, please remember, this presentation is approved for continuing education credits. Click on the free CE credits button in the bottom left-hand corner to obtain your credits for this presentation. I would now like to introduce today's speaker, Dr. Marilyn Brown. Dr. Brown is currently Corporate Vice President, Global Animal Welfare at Charles River. She is board certified in laboratory animal medicine with both ECLAM and ACLAM certificates. From 1982 to 1990, Dr. Brown directed a large experimental surgical facility at the University of Illinois, Chicago, where she also served as Associate Director of the Biologic Resources Laboratory. She was the Director of Dartmouth College Animal Care and Use Program and Assistant Professor in the Department of Microbiology at the Dartmouth Medical School from 1990 to 2002. While at Dartmouth, she served as the Chairperson the Faculty Advisory Board of the Ethics Institute. Dr. Brown has over 30 publications in various areas of laboratory animal care and use, including several chapters in the National Agricultural Library's publication, Essentials for Animal Research, and has served as an author and editor in the ACLAM book series. She served on the ASMA panel on research animal surgery and was on the ILAR committee to revise the Guide for the Care and Use of Laboratory Animals in 1996. She has been particularly interested in the areas of surgery, anesthesia, IACUC functions, humane endpoints, ethics, and animal welfare. I will now turn it over to Dr. Brown for her presentation.
Thank you, Brenda. I thank you for that wonderful introduction. It's practically as long as my talk is going to be. And I want to thank LabRoots and all the sponsors who've made this continuing education event possible. So the next slide will, oops. Okay. So this slide will describe a little bit about how I'm going to approach this topic. In the introduction, I'm going to briefly touch upon why it's so important that we pay attention to animal welfare. And there are regulatory, ethical, public relations, and scientific influences. Although stress is not my area of expertise, and I must admit I was a bit stressed in preparing for this presentation, so I'm a little more expert about it than I was before I started. I will mention some of the basic definitions, the sources of stress in the laboratory, and some mechanisms by which stress may impact on research and testing results, and thus decrease the reproducibility of animal studies. The last half of my talk is going to talk about strategies for minimizing stress in the laboratory. I'm going to conclude by referencing applicable ARRIVE guidelines in case you missed the earlier talk on promoting reproducibility yesterday or the keynote lecture on ARRIVE guidelines that occurred this morning. And I just got a notice in my email just this afternoon that the NCCR's ARRIVE guidelines are being featured in Nature just this week, so it's a very timely topic. So I'm sure that, are my slides all advancing as they should? Yes, okay, thank you. I'm sure that everyone in this audience is well aware of the various regulations and guidelines that serve as some of the basis for our attention to animal welfare. But I feel I have to include them here just for the sake of completeness. Basically, they cover really the same three key points. One is that procedures involving animals will avoid or minimize discomfort, distress, or pain to animals. Second, procedures that may cause more than momentary or slight pain or distress to animals will be performed with appropriate sedation, analgesia, or anesthesia unless withholding those treatments is scientifically justified and right. And lastly, that animals that would otherwise experience severe or chronic pain or distress that cannot be relieved will be painlessly euthanized at the end of the procedure or, if appropriate, during the procedure. So, of course, there's also ethical considerations for being concerned about animal welfare. When talking about morality, one often refers to the moral community, or who is part of that community. Referring to where someone fits in this community is referred to as moral status, moral standing, and/or moral considerations. I'm going to use just the phrase moral standing. Rowland considers the moral standing of animals to be the key question of veterinary ethics. It's easy to get caught up in definitions and differences of each of these. So, as I said, I'm going to just be using the moral standing. Why is moral standing important? Moral agents, that would be humans, have moral obligations to entities with moral standing. The view of moral standing comes from legal terminology, one's place in the community, in the estimation of others, one's relative position or social, commercial, or moral relations, one's repute, their grade, or their rank. Moral standing is usually ascribed to members of a group rather than an individual. So in other words, it would be all humans, or all monkeys, or all apes, but rather than just an individual monkey or ape. This definition seems to apply a continuum or sliding scale of moral status, since one may have a higher or lower position in social, commercial, or moral relations, and thus one may have a higher or lower moral standing. The concept of a sliding scale of moral standing is supported by Fry, who said that although animals have moral standing, animal life is less valuable than human life. Fry believes moral intuitions support this comparative value thesis. Moral intuitions are the relationship of morals to common sense, and are also one of the factors considered when ascribing moral status to infants or brain-damaged humans. Beauchamp's statement, animals have standing that protects against their being treated in certain ways that would reduce their lives to the level of things or mere resources for others. This also leads open the idea that such standing may be given on a sliding scale. Evolutionary theory, which lacks sharp breaks, also supports the idea of a sliding scale or continuum. The higher up one is in the evolutionary tree, the higher their moral standing may be. The question of moral standing of animals is not the same as the question if animals matter. Carruthers said common sense morality tells us that animals have partial moral standing in that their lives and experiences have direct moral significance, but much less than humans. Well, enough philosophy. And what about social science? What does the public think about animal welfare and science? This survey clearly indicates the public's interest in the welfare of animals. This was a Gallup poll based on telephone interviews conducted in May of 2015 with a random sample of adults 18 and older living throughout the United States. The percent of respondents who felt that animals have the same rights as people when it comes to protection from harm or exploitation is roughly half that of the percent that feels that animals deserve some protection. But it's interesting to note that this percentage for those that feel that the rights are the same is increasing. Although I don't have recent survey data for this, whenever I have seen information about the public, how the public feels about the use of animals in research, typically the level of support the public has goes down once the question is phrased that the particular involves pain and or distress. So clearly, it's the pain and or distress part of experiments that are of the greatest concern. Let's go across the pond and look at the UK. This graph represents the acceptance of animal experimentation in the UK, and is actually a composite of several questions. And so the participants could answer numerous questions, and they could answer them differently. So maybe questions 1 and 3, they said were conditionally accepted, and maybe question 2 was unconditional. So therefore, these numbers don't add up to 100, so I do realize that. But I think it's important to note that the unconditional acceptors that started at 35% in 1999, in 2012, it was 55%. So this does show some increase in people having unconditional, feeling unconditionally accepting of the use of animals in research. Next, I found this information actually which is from a Nature survey in 2011, where they surveyed they sent an email to 20,000 biologists and invited them to take part in this poll. And roughly 1,000 people did respond. I think it's interesting to note that the scientists responding in the U.S. and the UK, the very significant majority said they had no ethical concerns about the role that animals played in their research. This actually surprises me. This percent goes down when looking at continental Europe and the rest of the world. I think these results are of particular interest when we look at the next slide. Regardless of the level of ethical concern, the opinions about the essential role animals play in the advancement of biomedical research is really similar in all regions of the world. And lastly, all regions seem to agree that the legislation governing the use of animals in their specific region is about right. I think it would be very interesting to ask this question in the general public and see if we have the same answer. I've mentioned the regulatory, ethical, and public relation reasons we need to pay attention to animal welfare. But for the purposes of reproducibility, we need to look at the scientific impacts of animal welfare. Stress will result in catecholamine release, and that activates the hypothalamic pituitary adrenal, or HPA axis. This increases serum glucocorticoid concentrations. Downstream effects of these neuroendocrine signals may include decreased body weight or body weight gain, changes in food consumption and activity, altered organ weight, particularly the thymus, spleen, and adrenal, lymphocyte depletion in the thymus and spleen, altered circulating lymphocyte counts, such as increased neutrophils and decreased lymphocytes and eosinophils, increased serum glucose concentration, and altered reproductive function. One can only imagine how this can impair or influence studies that are in reproductive toxicology. In general, circulating leukocyte counts, lymphocyte subsets, and adrenal gland and thymus weight are the most sensitive parameters that indicate stress in tox studies, with the stress glucogram prominent in acute stress, and changes in total body weight and organ weight seen in more chronic stress. These stress effects need to be considered as secondary or indirect versus primary or direct test article-related findings. The impact of possible stress-induced effects on data interpretation can partially be mitigated by toxicology study designs that use appropriate control groups. So in other words, cohorts are treated with vehicles and subject to the same procedures as those dosed with the test article. It should also look at housing that minimizes isolation and offers experimental enrichment, and experimental procedures that minimize stress and sampling and analytical bias. Bias is due to order of collecting samples or taking measurements must be minimized by conducting procedures on animals across groups in either a random a round-robin stratified fashion. So what is stress? I probably should have started with this slide. So basically, it's been referred to as a physiologic response that an organism mounts to adapt to stressor to move towards homeostasis. It's also been referred to as general adaptation syndrome. We all know that there's something called eustress, which is good stress. This might be the stress that I may have when I have a good piece of chocolate cake put in front of me, versus distress, which is what I might feel when you took that piece of cake away, which is bad stress, an adversive state resulting from maladaptation or inability to adapt to stressors. Well, okay, maybe I could adapt to having you take the cake away. I think I'd last for quite a while without chocolate cake. Another term you might see is allostasis, which is a set of responses to normal, to restore normal physiologic conditions. Stress in a laboratory setting can be divided into 4 major buckets. The first is real-life situations. These are typical lab procedures such as transportation, husbandry, housing, handling, and some sample collection. Real-life stress responses tend to be transient and result in habituation within 1 to 4 days. The next bucket would be disease. The next would be experimental perturbations of the environment. They could be emotional or physical. And the fourth bucket would be pharmacologic or surgical induction of stress. Unfortunately, in my slide, I might have misrepresented this where I talk about dietary restriction and inappetence under just the pharmacologic induced stress, and I think that's primarily because I was, my filter in looking at this information was from a toxicology standpoint. But certainly dietary restriction or lack of appetite could occur in any type of stress, not just pharmacologic or surgical stress. Stress also can be divided as acute stress. Those might be the short dosing, restraint, even of up to a couple of hours, the short duration of test article effects, maybe such as vomiting immediately after dosing. Chronic stress is that which lasts for more than a few hours, or it may be a continuously applied stressor, an example being chronic social stress, or repeated intermittent application of acute stressors over a period of days or weeks. And these could be predictable or unpredictable, although the unpredictable stressors appear to be more difficult for an animal to adapt to. How can stress affect research results? Well, as I mentioned, you can see increased glucocorticoids. This could increase basal metabolic rate, and as you might guess, that would potentially alter metabolism of your test article. You might also see alterations in tissue perfusion. This potentially also could change metabolism of test article, or it could change the exposure of the active compounds of your test article to the targeted tissues. These impacts often affect organ systems other than the immune system, most prominently the endocrine and reproductive systems, as I mentioned earlier, and clinical pathology, primarily hematologic parameters. Sorry. So when you're looking at the results of a study and you're trying to assess if the findings are the result of stress or if they're primary toxicity from the test article, you want to use a weight of evidence approach. So one of the first things you're going to do is you're going to consider the biology of the test article and its intended target. You're going to evaluate the magnitude of the response in the stress-sensitive parameters and organs. You can examine the dose response of the potential stress-related changes versus the dose response of the probable primary test article. And usually what will happen is if there is a greater test article effect, there will also be a greater stress effect. So you want to consider other potential interpretations of your data. For example, lymphocyte depletion in the spleen or lymph nodes, but not in the thymus, which is generally a more sensitive organ to stress, may indicate that the effect is a primary effect of the test article versus an effect of just stress. You want to evaluate the strength of the weight of evidence. So generally, the less sensitive indicators of stress do not occur in the absence of the more sensitive indicators of stress. One of the things that you want to avoid is multi-system failure. So what are the sources of stress in toxicity studies? Well, of course, one of the things we mentioned is the primary test article effects. These can be particularly in the high dose groups, which may show severe clinical signs. They may be of a sufficient magnitude and or duration to result in stress as a secondary effect. Because primary effects of test article are generally proportional to the dose administration, the findings due to stress may also show dose proportionality. Minimizing pain and distress through veterinary intervention is of utmost importance and something that I'm going to be discussing in greater detail as I discuss humane endpoints. One way that you can minimize stress in the laboratory is through excellent training of your technicians. I was talking with some of our toxicologists and pathologists as I was preparing for this talk, and I found it interesting that the study directors and pathologists routinely stress the critical importance of the experience of the technicians. They need to be able to recognize subtle signs as well as more overt signs. They need to know what's normal, and usually the technicians do know what's normal in the animals that they see every day, and therefore they can recognize when something's abnormal. This is particularly important as when we talk about humane endpoints, that they recognize what types of clinical signs have been identified to be tied with earlier endpoints. They need to be proficient in the procedures that are carried out so that they're going to minimize the negative impact on animal welfare as well as impact variability. For example, if you have a technician where the animal is much less stressed when it's restrained for a blood collection, you're going to have more consistent results and less variability. They need to have an understanding of the procedures that are going to be followed and when adverse effects are expected to be seen. They need to know how to report those events and when to report them. And they need to have an understanding of the criteria that have been set for humane endpoints. This training needs to include not only your primary personnel, but clearly your backup personnel that are going to be covering on weekends and non-peak times. You can minimize stress by refining techniques. Acclimation is critical to decreasing stress to animals in the laboratory. Depending upon variables to be studied, transportation can be one of those stresses, and it can impact animals for up to several weeks, although the average time to reach equilibrium is 3 to 14 days. For routine tox studies, the modern practice of acclimating rodents for at least a week is usually adequate to recover from the stress of shipping. Isolating rats in novel cages to collect urine cortisone or corticosterone is transiently increases serum corticosterone by approximately 8-fold over baseline, but this increase returns to baseline after approximately 120 minutes. So this is just one example of where you want to acclimate the animals before you start collecting data. Habituation is measured by a routine to baseline of one or more stress-related parameters. Operant conditioning, in this particular case, this is an animal who was having a topical application of a test article, and this animal was trained to voluntarily walk up onto the top of the table where he got a treat as he had a stockinette put on over the test article to protect it from being rubbed off. So here's an animal that is voluntarily being involved in the actual dosing. You see this as well. It's very common in non-human primates to train the animals to voluntarily submit an arm or a leg for blood collection. Restraint can be associated with increases in heart rate, increases in blood pressure, and decreases in food consumption and weight loss. Non-human primates subjected to chair restraint, squeeze cages, or manual restraint have increases in serum and urine corticosterone concentrations for some time. Adaptation to daily restraints for nose-only exposure, as measured by normalization of body temperature and heart rate, can occur after just 2 weeks in rats and mice. Circulating catecholamine concentrations increase as soon as 10 seconds after a stressor, while increasing in corticoid concentrations are not detected until approximately 3 minutes after a stressor. Historically, blood collection by decapitation has been considered to be one of the least stressful methods in rodents. Several recent studies, however, suggest that the procedures involved in decapitation are a source of acute stress for rodents, and their extent may be substantial enough to impact several biochemical parameters. In rats, collection from the tail vein or from an indwelling catheter is appropriate for measuring ACTH and corticosteroids during stress studies, whereas decapitation after CO2 or pentobarbital causes stress-related effects on ACTH concentrations in trunk blood. Alterations in heart rate and blood pressure are lower in rats subjected to jugular bleeds when compared to tail vein or periorbital sampling, suggesting that jugular bleeding is less stressful. Collection of small volumes via previously implanted catheter in rats is also considered to be less stressful. Tail bleeding leads to increases in corticosteroid in mice immediately after bleeding, which is reversed within 24 hours. So there is species differences in their response to blood collection. In general, blood collection in large animals is associated with fewer stress-related changes compared to rodents. No difference in blood collection from cage-restrained dogs or dogs removed from their cages that are restrained in the anteroom have been seen. Stressors associated with housing include routine cleaning, housing density, movement of cage racks, and provision of environmental enrichment activities. In rats, routine cage changing, animal weighing, and other routine non-invasive procedures increase heart rate to a degree comparable to or even greater than parenteral injections or the odor of rat blood. Movement of cages of Balsi mice from one room to another results in changes in the HPA axis activation and decrease in circulated lymphocyte counts, with recovery within just a few days. Rats housed in enriched environments experience a state of eustress, as indicated by generally higher corticosteroid measurements relative to age-matched controls. Group house female rats exhibit decreased stress responses to routine cage manipulation as measured by heart rate and mean arterial blood pressure. Male rats subjected to crowding have decreased food consumption with concurrent decreased body weight gain and increased water intake. Isolated female rhesus monkeys have higher cortisol concentrations and a dampened circadian rhythm compared with stable social environment, animals in a stable social environment. But some other researchers have found no difference in singly housed versus co-housed rhesus monkeys. Dogs show less stereotypic behavior when socialized. Taken together, these data indicate that stress in animals placed in tox studies may be lessened by sufficient attention to housing and socialization needs. Types of cages, can affect body weight gain in rats after 3 days in a wire mesh cage. And this is only slightly less than the body weight of animals in solid bottom cages, which is, I think, somewhat contrary to what most people feel about wire bottom cages in rodents. As I mentioned earlier when I talked about the public's concern about the use of animals in research, and that the main factor that is of concern is that we want to be sure that pain and distress is minimized. We do this through analgesics, through veterinary interventions if animals are showing clinical signs, and in a moment I'll talk a little more about the OECD guidelines. It's essential that there be a team approach in any research setting, but particularly in a toxicology setting. The roles of the different individuals on this team, the study director, the veterinarian, and the technician, and the sponsor, all need to be clearly defined. And we need to assure that there's open communication between all parties, and that the parties who are making decisions, that there is someone always available to the technician should there be adverse events. When you're considering developing a intervention plan, the word plan is critical. You need to plan and plan and plan again. Have pre-study meetings so that everyone is on the same page. You might want to consider for each particular study a veterinary intervention plan. This could include how communication will occur, both urgent and non-urgent. You might look at what the test and control article effects are, what's expected. What are the safety concerns? What has exploratory data studies shown that you might expect? What about the PK data? What treatments are not allowed, and what treatments in, say, the pre-study phase, and what treatments are or are not allowed during the dosing phase. And you may also want to consider to have a document that includes the common substances or the common spontaneous conditions that you might see in that given species and how they will be handled, as well as commonly used drugs, so that the sponsor and the veterinarian and study director can all identify those particular agents which may or may not be appropriate in a given study. Of critical importance to animal welfare and the quality of scientific results, regulatory compliance, and the maintenance of the public's support of animal research is the assurance that pain and distress is minimized. As previously mentioned, animal welfare is stressed in various domestic and international regulations and guidelines. When it comes to recognition, assessment, and use of clinical science of humane endpoints in tox studies, the OECD has created guidance document number 19 in the series on testing and assessment. The purpose of the guidance document is to apply the principles of the 3 R's to the use of animals in regulatory toxicity testing. The ultimate purpose of the application of humane endpoints is to be able to accurately predict severe pain, severe distress and suffering, or impending death before the animal actually experiences these effects. It requires careful clinical examination using well-defined endpoints and criteria. Adverse conditions, once identified, should be minimized or eliminated either by humanely killing the animal or in long-term studies, perhaps by temporary termination of exposure or reduction of test substance dose, and or supportive care. If relevant humane endpoints have been identified, they should be described when an experiment is being planned and incorporated into the experimental protocol and all related SOPs, and of course approved by the IAPIC or appropriate oversight body. You might want to look at things like body temperatures. Do they increase? Are they expected to increase or decrease? These are often very sensitive endpoints. Other biomarkers, such as platelet counts, looking at disseminated intravascular coagulation, may be other things that you look at. Study protocols should clearly define the conditions under which it's necessary to immediately and humanely kill an animal or provide veterinary intervention. You want to avoid the moribund state or even more, an animal being found dead with the result autolysis of or autolysis of tissue and loss of the very valuable data. The OECD guidelines encourages the use of preliminary or range-finding studies and urges that these be used to obtain information which can be then used to identify earlier endpoints in the full study, which can be used later. It stresses the importance of adequate training, which we've already talked about, on the recognition and assessment of clinical signs. And it has a wonderful appendix that provides examples of these clinical signs. It recommends the use of checklists, stating that some of the advantages includes that the sign, if you just click if the sign is either present or absent, it minimizes observer error. It lists observation likely to occur or considered critical, so they're not overlooked or unambiguously recorded. It improves observational skills with staff training, and it must allow that other observations which haven't been anticipated can still be recorded. The OECD guidelines also, as I said, provide signs and conditions which may be indicative of impending death and or moribund conditions, so that these can be used to determine a humane endpoint. It provides signs which may be indicative that an animal has experienced significant pain or distress. It provides guidance on the humane conduct for the specific types of toxicity studies such as single dose or ocular studies. It has an annex that includes questions to determine whether earlier endpoints are possible and have they been sought. And it also provides an annex of clinical signs or conditions which indicate the need for closer observation or humane killing. Okay, so to the ARRIVE guidelines, I just really looked at the ARRIVE guidelines as they related to the materials and methods rather than say, statistical analysis and other aspects of the study. So the things that we talked about today, how the animals are allocated and into treatment groups, the experimental procedures, how are they done? When, what time of the day do they occur? Where do they occur? Do they occur in the animal's cage? Do they occur in an anteroom? And why? Experimental animals, the source, the correct nomenclature, of course, goes without saying, the health status of the animals, and any previous procedures that those animals may have had. Housing and husbandry details, including the type of facility. Is it an SPF facility? Is it a conventional facility? Are the animals perhaps exposed to other species besides their conspecifics? The type of caging, is it wire, is it solid bottom, how big is the caging, what type of bedding is used, what type of environmental enrichment is being used, what do you have for socialization? Are they pair housed, singly housed? If they're singly housed, do they have any opportunity for interaction with conspecifics, say maybe through a grooming bar or at least being able to see conspecifics? What is the light dark cycle? And of course, what is the nutritional status and water source? Welfare-related assessments and intervention should be included so that if you're doing assessments at a certain frequency, that should be included so that if someone is trying to repeat the study or reproduce the study, that they are using those same time intervals for their assessment. And that the interventions are generally the same, although we do have to allow for the fact that some animals, individual animals, may respond differently, so you may need additional interventions. What is the sample size, and of course, what are the experimental outcomes? So I'm a little early, but I want to show this particular poster, which the NIH came out with a long time ago, and it's just as true today. The NIH created this poster, which is good animal care and good science go hand in hand. So thank you very much, and with that, we can open it up to questions.
Thank you very much for that presentation, Dr. Brown. That's important information to bring to us. Before we get started on questions from our attendees, I'd like to remind the audience how they can submit their comments, questions, or answers. They can be submitted through the green Q&A button at the lower left. We're going to try to get to all of the questions today, but if not, we definitely can follow up after the broadcast. It's also important to remember that this presentation has been approved for CE credits. If you find the Get Your Free CE Credits button in the lower left, that will tell you how to go about earning credits for today's presentation. Now, I just want to give our audience just a few more minutes to get in their questions and see what we have from our audience today. It's really very interesting.
Make you tired.
We've had a great conference. We've had so much here today that's been really valuable. And I like this graphic at the end of your presentation. It really is very important to know that it's not all about the science. The animals matter too.
It might be a good ending to the whole conference, right?
I agree. Absolutely. Absolutely. We've had several speakers today that have talked about animal welfare and taking care of the animals that do so much for science. Okay. Now I'm looking in our question queue. I don't see anything right now. Do you have any final comments or thoughts about this topic for our audience?
Well, one thing I would just I would urge people to maybe take a look at the that OECD guidelines number 19. If they're at all involved, they probably may already be aware of it. I probably can't emphasize enough the importance of a team approach, that the animal care technicians at the case level are as important as the study directors, as the veterinarians, as the sponsors. It has to be a team approach in order for it to be, and in order for it to optimize animal welfare. I would also say what I will be doing is trying to create a bibliography. There were a few references that I think are particularly full of good information in this area, and so I can provide that to lab groups after the lecture.
Great. Thank you so much. We did have one incoming question. Well, not a question, a comment. Thank you for a great presentation and very useful information. So that's good to hear. I'm glad that the audience is enjoying that. Now, if we weren't able to speak with you today, we'll be sure and follow up with you via email. Thanks for taking part. We also want to thank Dr. Brown, our presenter, for joining us here at LabRoots, leading scientific social networking site. Please visit us at www.labroots.com. Today's webcast will be available for on-demand viewing on our website through August of 2016. And we'll let you know when it's been posted, and hopefully you can pass that information on to any colleagues who couldn't take part in today's live broadcast. Don't forget about your free CE credits in the bottom left corner. Thank you for joining us today. We'll see you again. Goodbye.
Thanks, Brenda. Bye-bye.